So here we are examining the process of gene expression, or how genes manifest themselves. We've examined transcription, which is how we get the messenger RNA copy from the gene itself. Now we're going to look at the process of translation. All the elements needed to synthesize the protein need to be brought together on the ribosomes. In this figure on the right, we have all the elements necessary. We've got the large ribosomal subunit, the small ribosomal subunit, a piece of messenger RNA, some transfer RNAs carrying amino acids. So all of these things need to come together. We're going to break this basically into 5 stages. We're going to have initiation, elongation, and termination. And then after we've had termination, after the polypeptide molecule is made, we then need to have protein folding and protein processing. So let's take a look at the initiation of translation. First of all, we have to have the messenger RNA molecule leave the DNA transcription site, and then that messenger RNA will be transported to the ribosomes that are in the cytoplasm. The ribosomal subunits are adapted for assembling and forming sites to hold the messenger RNA and tRNAs. So first, the smaller ribosomal subunit comes along, and then we'll have the messenger RNA log itself into the initiation site. And then the large subunit will plunk itself down on top, and now the whole thing is assembled and ready to go. Let's take a look at a quick animation of the initiation of translation.
Translation initiation is a process in which mRNA, initiator tRNA, and small and large ribosomal subunits associate with each other to form a complex. Initiation of translation in bacteria begins when the 30S or small ribosomal subunit binds to the mRNA near its 5' end. This process is facilitated by a Shine-Dalgarno sequence in the mRNA, which is complementary to a component of the small ribosomal subunit called 16S ribosomal RNA. Though a shorter sequence is shown here, the Shine-Dalgarno sequence is actually 9 nucleotides long. Initiation factor 3 also facilitates the binding of the mRNA to the small ribosomal subunit. Once the 30S subunit IF3 mRNA complex has formed, initiation factor 2 binds to the complex and promotes the binding of the initiator transfer RNA for N-formyl methionine to the complex. IF2 and IF3 hydrolyze GTP as an energy source to promote the association of mRNA, tRNA, and the ribosomal subunits. Translation initiation is completed when the large ribosomal subunit binds and IF2 and IF3 are released. Later, when translation is completed, IF1 is needed for the dissociation of the complex.
So you don't need all the details of the initiation factors and such, but hopefully that gives you an understanding of how it all comes together. Before we start looking at the elongation phase, we better get a better understanding of the genetic code, the message that's in the messenger RNA. The genetic code is simply the messenger RNA codons and the amino acids that they specify. In this table below, you can see the various codons on the messenger RNA and each of the amino acids that they code for. You'll notice that there's one start codon, and then there are 3 different stop codons. The start codon is always AUG, and so the first amino acid in any polypeptide chain will always be methionine. However, methionine often gets cleaved off during protein processing. You'll notice that most amino acids have at least one codon that codes for them. Some of them have even more. So there is redundancy in the genetic code. That is, there are many different ways to say, for example, leucine. Leucine has 6 different ways that you could say it. You could say leucine with UUA, UUG, CUU, CUC, CUA, or CUG. It doesn't really matter. Either way, that codon is going to code for a transfer RNA that's carrying leucine. Because of this, it's evident that only the first two nucleotides are often required to encode the correct amino acid. In this case, all of these begin with Cu. So if a Cu is there, then we're definitely going to get leucine. This is thought to permit some variation in the DNA without actually altering the message. So this is not a table that you need to memorize or anything, just understanding that the codons on the RNA that are shown in this table call for a transfer RNA that brings the corresponding amino acid to the growing polypeptide chain. So how was it ever discovered that this code was read in triplets to start with? Well, they recognized that there were 20 different amino acids and only 4 different possible letters in RNA, A, U, G, and C. And how could you possibly code for 20 different amino acids with only 4 different letters of the alphabet to do it? So then they thought, well, maybe a 2-letter code. Well, if we have 4 letters squared, then we end up with 16, which isn't quite enough to cover all 20 amino acids. So then they supposed that perhaps there was a 3-letter code. And if there was a 3-letter code, we could have 64 possible different codons. And that's exactly what you're seeing in this table. 64 different codons that cover 20 amino acids. That's precisely why there's some redundancy. So again, to review how the message gets from the DNA, we have the template strand, which actually has the relevant information on it. And when that's transcribed, it produces a messenger RNA. The messenger RNA has codons on it. And then the transfer RNA comes along, and it has a matching anticodon that links up with the codon on the messenger RNA. And each tRNA with its specific anticodon is carrying a specific amino acid. As those amino acids are brought into the ribosome, they're added to one another in order to create a polypeptide chain. So now we can look at the elongation process in the polypeptide. This is the figure that your text gives you, showing you how the pieces come together. However, I prefer this figure. We began at the initiation phase here. We have the ribosome assembled on a piece of messenger RNA with the first tRNA in its slot carrying methionine. The next stage in the game is for another transfer RNA to come in reading this codon and bring a corresponding amino acid. What happens here is with the fuel of some GTP, we're able to take the 2 amino acids and stick them together. So the amino acids from here pop over onto this transfer RNA, and now we have a chain right here. Now the ribosome moves along the messenger RNA strand one more step, creating space for another transfer RNA and its corresponding amino acid to arrive in the A site, or the arrival site. It arrives, energy comes in, and then we can move the amino acid chain over again. This elongation cycle continues until we reach a termination site. Let's take a look at a quick animation of how elongation occurs. Motion here is really worth a million words.
Chain elongation begins with the binding of a tRNA, which recognizes the next codon in the mRNA to the A site of the ribosome. This is catalyzed by the EFTU transcription factor and requires the hydrolysis of a GTP. Once the tRNA binds in the A site of the ribosome, the polypeptide chain is moved from the tRNA in the P site to the amino acid attached to the tRNA in the A site. Peptidyltransferase, a protein RNA complex present in the 50S ribosomal subunit, catalyzes the formation of this new peptide bond between the amino acids. The ribosome then translocates to the next codon. This process is promoted by elongation factor G and requires another GTP. This places the empty tRNA molecule in the E site of the ribosome and moves the tRNA containing the growing polypeptide chain in the P site. The next codon in the mRNA chain is positioned in the A site. The uncharged or empty tRNA in the E site then leaves the ribosome and a cycle of chain elongation is completed. Through subsequent cycles of chain elongation, the polypeptide chain continues to elongate one amino acid at a time.
Hopefully that helps in your understanding. Again, you don't need to know the names of the elongation factors. I remember these sites as A, P, and E as arrival, polypeptide, and exit sites. So now let's look at the final portion of translation proper. Keep in mind we still have to fold the protein and process it before it's good to go. So we've had our transcription, we've translated the protein, and now we've reached the end of the messenger RNA segment. How does it all end? This is brought about by one of those termination codons. Remember, there were 3. You don't have to memorize them. They're often called nonsense codons because they don't code for a transfer RNA at all. Instead, they code for a release factor. When the release factor lands in the A site, essentially the whole thing blows apart. It involves a special enzyme that breaks the bond between the final tRNA and the finished polypeptide chain, and thus releasing it from the ribosome. Let's take a quick look at an animation of the termination process.
Termination begins when a stop codon appears in the A site. Since there is no tRNA corresponding to the stop codon, a release factor binds in the A site. The binding of the release factor causes the polypeptide chain to be cleaved from the tRNA. The polypeptide is released, and then the tRNA is released. In the last step, the 2 ribosomal subunits and the mRNA dissociate from each other. This completes the termination process.
Before the polypeptide is really ready, it actually has to become a protein. It's going to become a protein by becoming modified. There's many post-translational modifications that are necessary. Before it's released from the ribosome, the polypeptide chain will start to fold upon itself and achieve its tertiary conformation. You'll recall from that chapter earlier in the semester. Often we're going to clip off the first amino acid because it's methionine, and not every polypeptide chain needs that methionine. Sometimes we'll add cofactors, and sometimes those polypeptide chains with their tertiary structure will need to join other tertiary structure proteins in order to assume their quaternary structure. So the process of transcription and translation is really efficient in prokaryotes. So efficient that actually ribosomes are able to assemble on this messenger RNA transcript as it's being made. Before we move on, we'll have one final animation that should summarize the whole process of protein synthesis.
Translation is initiated by formation of an initiation complex consisting of the smaller ribosomal subunit, the first amino acid tRNA, and messenger RNA. The larger ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has 2 sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. The initiating transfer RNA, carrying its amino acid, binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site. The ribosome now advances a distance of 1 codon and the first transfer RNA is released. A transfer RNA carrying the next amino acid now moves into the A site and the ribosome shifts down by a distance of 1 codon. This is followed by the 2 amino acids on the transfer RNA in the P site being transferred to the third amino acid. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when the ribosome reaches a codon that does not code for an amino acid, called a stop codon. The ribosome dissociates into the smaller and larger subunits, and the messenger RNA and protein are released.
So there you go. That's the level of which you need to understand protein synthesis for this course. Now we can move on to see how it all happens in eukaryotes.